Good morning, respected judges. My name is Saransh Handa, and I'm an undergrad student. So, the topic that I've chosen for my poster presentation is salivary biomarkers and oral cancers. So, what interests me about this topic is that oral cancer has a very high prevalence rate in a country like ours, right? So, there are 30,000 new cases every year, and the 5 year survival rate is less than 50% as I've mentioned it here, right? So why is there a need of salivary biomarkers? So when there is a delayed detection, which is usually the case, it leads to high death rates in a country like ours. So salivary biomarkers are basically needed because of the early detection in cases of oral cancer, such as squamous cell carcinoma. So, before moving on to the relationship between these two, let us talk about the definition of a salivary biomarker. So, a biomarker, what is it? It is basically a biological molecule that is found in body fluids, tissues that have an objective that indicates the medical state of a person from the outside. And this can be measured pretty accurately and we can reproduce it. That is why we need biomarkers for detection of any condition, not just oral cancer. But why do we use salivary biomarkers only in specific? The answer to this question is because of its non invasive nature, firstly. Secondly, it's very easy to collect from a patient, right? And also, it is easy to transport and stored. So, some of the most common biomarkers are tumor DNA, as I've mentioned it here. Secondly, we have extracellular vesicles. Then we have microRNA, we have cytokines, and we have chemokines. So, the role of these salivary biomarkers is explained with the help of this flow chart. So, in easy language, how does this basically work is that the protein expression in the saliva is altered when there is presence of oral cancer, right? And this can be detected using mascot technique. And this technique, what it basically comprises of is that we can compare the protein sample, I mean the saliva sample of the patient and with the mascot sequence, right? And this is used to identify all the differentially expressed protein in cases of cancerous and pre-malignant lesions of the oral cavity. And we also know that salivary IgA levels and amylase, potassium levels, these are usually significantly lowered in case of oral cancer. Whereas calcium, magnesium, phosphate, sodium, all these levels are higher. And this can be studied and this can be verified with the help of studying the population by collection of the saliva. So the changes in their protein expression is noted by protein identification firstly, which is further followed by the mass spectrometry. So you may ask, what is mass spectrometry? So in this method, we derive the protein from a complex biomolecule, right? And then we can measure the charge is to mass ratio, which further leads to the verification of these proteins. And we know whenever there is an alteration in these proteins, it is a condition of oral cancer, right? So the verification of these proteins is done by salivary biomarkers. And this can further be confirmed with the help of various techniques like rapid ELISA and Western blot techniques, which is further compared with the histopathology, and these pathological changes can be studied under a microscope, which tells us what case of oral cancer it is, which stage we are at, and it usually helps in early detection, as I've previously mentioned. So, this is what the entire poster is about, the relationship between the two. I've also attached a picture right here of oral cancer. Thank you so much.